Welcome back, students. This is Module 4.2.5, Applications of Biotechnology. One of the major applications is in biomedicine, and so the medical applications are huge in terms of the potential. One of the really large areas would be pharmaceuticals. The future of medicine would be one in which we use molecules that are found naturally in the body rather than using foreign chemicals with many side effects. Sometimes you hear these advertisements for some drug and the side effects are so terrible, the cure seems much worse than the disease. And so the hope is that in the future pharmaceuticals will be made naturally by the body. But one way that we can genetically engineer and correct any problems is through biotechnology. And so we can make all sorts of proteins. We can, as we make insulin now, which is injected, HGH is human growth hormone. Gamma interferon was one of the first genetically engineered products. It was made quite a while ago, back in the 1980s, or maybe even, well, the 1980s anyway. And this protects human cells from viruses of various types that can cause diseases like hepatitis and herpes. Of course, we know that, as I explained to you earlier with the case of human insulin, that bacteria can be made into little factories, protein factories. And so if we insert the desired gene into a bacterium, it will produce the protein that we need. Of course, a major application under medical applications beyond just pharmaceuticals would be gene therapy. This is the alteration of genes to help correct inherited genetic disorders. Now, there is an issue here. We must be able to get a gene that we've repaired using gene splicing or genetic engineering. We must have a way to introduce it into the body. Here's a little cartoon that shows how we might inject or how we might correct a problem such as hemophilia, where the precursors to red blood cells in the bone marrow that produce a clotting factor are made, and how we could possibly have this corrected. So if you look here, here's a gene, and it's corrected, it's cloned, and we insert it into a retrovirus. So retroviruses can be used as a vector. We spoke earlier of bacterial plasmids being a vector, but a retrovirus can also serve as a vector. And so it says retrovirus capsid. That's the protein capsule around the outside. That's the viral DNA, and we can see our little gene with the correct clotting factors there. And so we could produce many retroviruses with the cloned gene in them, and then we would allow the retrovirus to infect bone marrow cells that have been removed from the patient and cultured. And so we would then grow many of these cells in tissue culture, and so the viral DNA carrying the normal allele would be inserted into the chromosome of those cells. So if we look here, we have the corrected gene that was placed by the virus into the nucleus. So we're using the virus to insert the gene into the cells, which are then grown in culture, and then when we collect enough, we can inject those cells into the bone marrow, and from then on, the body would produce those cells. They would be supported by the body, and they would produce normal clotting factor, and this would solve the problem of hemophilia. At least that's how we draw it on paper. There are certainly roadblocks. It's not that simple. We haven't been able to solve all the problems, but certainly the potential is there. These are mice of similar age that have received similar amounts of food, but the one on the right has received a gene for growth hormone, allowing it to be bigger. There are a number of conditions, and here's a table that lists a number of them that could be helped with gene therapy. And there are clinical trials, but the people who volunteer for clinical trials are often sick, often terminally ill, and as a consequence they often die, and there have been lawsuits that have slowed down some of these things. When someone dies, sometimes their family just wants to blame someone. Anyway, biomedicine has great potential for the application of biotechnology, and we'll continue to see it in this arena. A second area where biotechnology can be applied is agriculture. For example, we can develop plants, crop plants that have a gene for resistance to a particular herbicide. Herbicides kill plants, and they would be used. We could spray an entire field, an entire agricultural field, if all the crops that we wanted to keep were resistant to the herbicide. And so this actually solves a real problem. So it would kill the weeds, but not the crop, if the crop had the gene for resistance. We can also use genes because we have a problem with insects evolving resistance to insecticides. This is a huge problem in agriculture, and the use of biotechnology would allow us to overcome this. We could change both the insects or the production of insecticide in such a way as to overcome this. We could nitrogen fixation. You may or may not know that the atmosphere is 78% nitrogen, but it's a form that plants can't use, and nitrogen is one of the major limiting nutrients of plant growth. And when we make fertilizers, one of the most common things that's in fertilizer is nitrogen. And so there are, in fact, plants that have on their roots little bacteria, and the bacteria can perform this magical trick of turning the nitrogen from the air into a form of nitrogen useful for fertilizer. Now we can take the gene from that bacterium and put it into plants so that plants can make their own fertilizer, and this could greatly decrease the use of fertilizers. We can also use biotechnology to make foods more nutritional. For example, rice is a staple food for billions of people on the planet, and we have a global shortage of iron and vitamin A. The lack of vitamin A causes blindness in millions of children. Typically a child that becomes blind because of lack of vitamin A, they all often die within a year or two because they're living in abject poverty anyway. And so it's just a downward spiral. So if we could fix this, and the lack of iron causes anemia, and in fact, we have done this. If we look right here on this particular slide, it shows this is how golden rice was made. The reason it's called golden rice is because we took an enzyme for beta-carotene, and this word might look familiar as in carotenoid, accessory pigments. This is a yellowish color gene, but it's beta-carotene is also a precursor to vitamin A, and we put that gene into just regular old white rice, and it also turns the rice plants kind of a yellow color, and hence the name golden rice. We also put other genes. This is a combination of genes we put a gene from beans, we put a gene from a fungus, we put a gene from wild rice, and all these helped us to make rice more nutritious in terms of iron. One gene increases the iron content of rice. Another gene that will help the absorption is produced, and yet another one produces more sulfur, and sulfur helps iron to be produced. But don't memorize any of these details. Just know that it's one example. This is an example of a previous point that I made. These are plants. The two on the top have been treated with a gene for herbicide resistance. The ones below didn't have the gene for herbicide resistance. They were all sprayed with an herbicide, and you can see that some survive fine, the ones that have the gene. So there you go. Let's leave agricultural applications. There are certainly many more. We can make plants more salt tolerant or more water tolerant. by moving genes from one plant to another. But let's consider some of the ethical issues of biotechnology. And I'll do these by category. The first category considers biotechnology in industry. The biotechnology revolution is really the first major scientific breakthrough that has been made in a substantial part in private industry rather than in, for example, public universities or something like that. And so much of biotechnology and its data has been privatized. And so always the issue in the foreground is the desire to bring products to market and to make money. And much less attention is paid to the background issues, such as safety or ethical concerns and environmental issues. So that's certainly one major concern. And we saw some of these concerns addressed in the video Cracking the Code, in which they talked about a shift in the ethos of biological research and a compromise of communitarian values, that kind of thing. And that's when they were patenting human genes and for private companies, and that certainly causes concern. A second problem in the arena of ethical issues is eugenics. Now, the definition of eugenics is the systematic attempt to create a more perfect human by controlling the genetic material, that is, namely DNA. Now, there are two types of eugenics. Negative eugenics, although it sounds like it's negative, it's the attempt to eliminate genetically inherited disease. And almost everyone's in favor of negative eugenics. Very few people are in favor of genetic disease. On the other hand, what is considered positive eugenics is the selection of desired traits. And this is extremely controversial. Because what is a desired trait? Well, we make a bigger gap between the rich and the poor, as the rich are genetically engineered to become more intelligent or more capable, and the poor become separated. And of course, there's great concerns because eugenics is often associated with World War II and the Nazis and Adolf Hitler and the master race, the Aryan race. And so concerns of racism and other similar issues are attached to the concept of eugenics. Let us also consider a third point, and that would be environmental concerns. The history of human interaction with the natural environment has been one of unintended consequences. And when we change through recombinant DNA or gene splicing the genetic makeup of an organism, we create something that's called a GMO, a genetically modified organism. That's a GMO. And if we release a GMO into the environment, it could have devastating consequences. For example, what sounds good on paper is one of the earliest genetically modified organisms, one of the earliest GMOs, was a bacterium that could eat oil. Now just think of like the Exxon Valdez. It, of course, had a huge oil spill up in Alaska, and was very destructive and took much effort to try to clean up. If we could just release this genetically modified bacterium, and it would go in there. I think of them as like a little Pac-Man going wah, 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 wah. Of course, that's not what they are. It's just a bacterium that has an enzyme that allows it to digest the carbon chains that are found in oil, which is really what petroleum is, just hydrocarbons. But consider where oil spills typically occur. They occur in areas where oil is produced. Now think what would happen if a genetically modified bacterium with the ability to digest oil got into oil well, into an oil field. It could quickly consume all of the oil that's there, and we would be out of luck. And so that has never been used. That particular GMO has never been released. But it shows you the kind of concern. Also, we can put genes for pesticide production right into crop plants. And that seems like a good idea if your crop plant is wind pollinated. But what happens if you have a field of wind pollinated plants next to a field of insect pollinated plants, and the genes for pesticide production get into the insect pollinated plants, then if a pollinator insect comes onto that plant, it'll be killed by the pesticides. And so that would not be a good thing. And the genes in plants actually can move around a little bit. And so that's certainly an issue. A fourth concern under the topic of ethical issues is intellectual property rights. To whom does your genetic structure belong? Consider the patenting of human genes, or consider the case of a cancer patient who had a form of leukemia. His name was John Moore, and he was treated at UCLA Medical Center in California. During his treatment, his spleen was removed, and some unusual T lymphocytes, the cells from the immune system, were harvested and cultured, and it was believed that they could have great impact in treating cancer. Now the patient, so the regents of the University of California patented those cells and didn't tell the patient, John Moore. Later, when he found out, he sued for a share of the profits. Now, initially, he prevailed, but upon appeal, the California Supreme Court found that John Moore didn't have possession of his own genetic code, that the cells had negligible value while in his possession, and they only became valuable when the university developed them. And so that's quite a frightening spectacle to consider, for sure. Let's look at one more, and that is the concept of disclosure, and that is who has a right to know your genetic code. And this one example here was when a worker for one of the railroads had a health test, and in that test, he during that health examination, he received a blood test, and they found that in some of his genes, because you can get your entire genetic code from your blood cells, they found that he had some of the markers associated with heart disease, even though he had not manifested any heart disease. As a consequence, he was fired from his job. He sued and he prevailed, but it just shows the way that this information can be used. And you have to know that insurance companies hire some of the best geneticists around. They want to know because their profits are riding on this. And even with this disclosure, there remains a gap between what is ethical and what is legal, and so it is a concern. And so there you go. These have just been a brief sampling of some of those issues that will confront us as we manipulate DNA, as we participate in biotechnology. And once that box is opened, we cannot again close it because we're uncomfortable with the information that we get. And it is a two-edged sword. This information can be used to help, but it can be used to harm. And so there's, I guess, Pandora opening her box. And so that will conclude our module.